Hello, everybody. First of all, let me apologize if my dogs start barking. Apparently, today is their day to bark at squirrels outside the window. But, um, guys, today you guys have started talking about genetics, and today was about mutations. Now, I know there was the reading that looks like this that talked about substitution, insertion, deletion, and frame shift, and then you guys were responsible to not that. Where'd it go? To work on the worksheet that looks like this. Now, for some of you, I understand this is probably very, very confusing. Hence, I'm making this extra resource with these notes and this video to follow. Okay. So, mutations intro. The first one I'd like to talk about is substitution. This is exactly what it sounds like. It is just subbing one base for another base. So if you look at our DNA sequence down here, we have AAT, but instead, in our substitution, our mutation, we now have AGT. Okay? One thing to take notice, this is not shift the whole amino acid sequence. Now, what this is meaning is that it only affects that one A turns into a G. This GTG over here is still the same GTG that's down here. And the CCG that's right here is the same CCG that's right there. Okay? Meaning it's only affecting one sequence of the codon, not the other two. Okay? That is substitution. Just substituting one amino acid for another one. Okay? Now, the next one is called insertion. Now, insertion is inserting one base into something. Okay? Now, this can be two bases, this can be three bases. Typically, it is only one base that is inserted. Now, this is typically harmful because it shifts the whole amino acid sequence. So originally, if you guys can picture this, I'll even draw it here. Oop, wrong side. Originally, it went. Ooh, there we go. It went just AAT. Okay, and whatever that codon codes for, it was AAT. Cool. Okay, or AA or be UUA in your case for mRNA. Then there was also that three sequence. There's that three sequence. There was that three sequence. That three sequence, and then it stopped. Okay. Now, by inserting these two right here, we are actually shifting all of these other sequences, making that three pair, that codon, now be changed. That is going to cause problems when it comes to translation and transcription and ultimately making our proteins. So now these mutations are going to cause errors. There are problems. The next one is called deletion. And this one is where one base is removed, and it's also typically harmful because it shifts the whole amino acid sequence once again. So, if you look here, we have our DNA sequence. Nice. It's all paired up in threes. Those are your codons. Okay. If we take three out, the ones that were originally right here, now we're squishing together. So whatever that amino acid that was supposed to be there, maybe that was a stop codon, maybe that was a start codon. What was ever supposed to be there is now gone. So the protein that we were supposed to make no longer works perfectly. Okay? So if you look at the image over here in the right corner, deletion can cause your chromosome to become shorter. That means you're missing a part of your DNA sequence, which means that your proteins are not going to be able to fully form, which can cause a lot of issues. Okay? That's deletion. Now, how do mutations occur, guys? There's a lot of different ways this can happen, but it's substitution, insertion, and or deletion. So there's three types, which is just going to affect the protein folding. And we were going to talk about the protein folding a little bit, just how it happens. So we have hydrophobic and we have hydrophilic. Guys, remember that hydrophobic means that it's scared of water, and hydrophilic meaning that it loves water. Okay, so little gray parts are hydrophobic, the little blue parts are hydrophilic. What happens here is that those hydrophobic parts actually bond to each other, and the hydrophilic parts don't. But if we switch these represent amino acids, now our protein's going to fold differently, and folding corresponds to the function of the protein. Okay, think of it if you have a big ball of yarn. You can knit and crochet into anything, but if you start misfolding, you might not make your gloves anymore. You might make a scarf instead. Okay, that's going to cause problems, as it's not exactly what you wanted. Now, guys, I know this is not perfect, but I just want to give you guys a little bit of extra resource and be able to show you what substitution, insertion, and deletion is. I'm going to go through one example on this worksheet quick, just so you guys can kind of see it. So if you did watch this video, good job. You're gonna get a little bit of help. So it says original DNA sequence, and it's given to you right here. And it says underneath you need to do the mRNA sequence, and then you need to do the amino acid sequence. So earlier you guys got a codon wheel. You're still gonna need that codon wheel. Okay? That's the truth behind the matter. You're gonna need it. So the mRNA sequence is gonna be T is gonna bond with A. A is going to bond with U, and C is going to bond with G. Bam. Now you're going to put those three letters into your codon wheel or your codon square, whichever one you guys have, and you're going to write the amino acid sequence underneath. I do have the answer key to this assignment actually linked below. Okay, so if you scroll all the way down, it's right there for you. Okay. The reason for this, guys, I want you guys to be able to see what mistakes you're making and be able to learn from them. Okay. So in our case, AUG, which we did, is in a code for MET, M-E-T. Okay. If you have any other questions, please feel free to ask, email me, anything like that. We'll get it done. Other than that, guys, have a good day. Thank you, and talk to you later.